Assalamu alaikum everyone. Today I'm gonna discuss about PCR. First of all, अगर हम इसके इंट्रोडक्शन की बात करें, PCR is a powerful nucleic acid amplification tool with exponential amplification efficiency, which can amplify a single piece of DNA in thousands of pieces within a short period. Basically, PCR technique used to amplify a specific DNA sequence. It works by heating and cooling a reaction mixture multiple times. This process allows for rapid production of millions of copies of DNA segment. Its invention by Mullis in 1984. Next, अगर हम components of PCR की बात करें, a PCR machine needs a DNA template, primer, DNA polymerase, nucleotide, and buffer solution to function. The DNA template includes target sequence that will be amplified. Primers are short, single-stranded DNA sequence that flank the target region and serve as starting point for DNA synthesis. Next, अगर हम इसके principle की बात करें, PCR work by heating the DNA to separate its strand. Then cooling it so primer can bind, and finally extended the DNA strand with tag polymerase to create multiple copies. Next, अगर हम इसके प्रोसीजर की बात करें, PCR amplifies a DNA segment. In denaturation, DNA is heated to separate the strand. In annealing, the temperature is lowered and primer bind to specific sequence of interest. Then in extension, in tag polymerase extend the primer, creating the copies of target DNA. Then repeat the cycle, continue amplifying the target DNA exponentially. Next, अगर हम इसकी types की बात करें, nested PCR improve the sensitivity and specificity. Multiplex PCR, it amplify multiple targets simultaneously. Then quantitative PCR, it quantifies a non-DNA sequences. Arbitrary prime PCR, a DNA fingerprinting technique. Then traditional PCR, standard PCR used for amplification of DNA sequences. Real-time PCR, basically it allows for quantitative measurement of DNA in real time. Then reverse transcription PCR, basically हम इसे RT-PCR भी कहते हैं. It is used to amplify DNA sequences by first converting them into complementary DNA. Then अगर हम इसके applications की बात करें, PCR has revolutionized fields like forensic science, research and genetics. In forensic science, PCR is used for DNA profiling and identification to help solve the crimes. In research and genetics, PCR is used for tasks such as genome comparison, phylogenetic analysis, gene expression analysis, and gene mapping. In medicine and diagnostic, tissue typing for organ transplant, personalized cancer treatment, early detection of HIV and other infections, bacterial contamination testing in blood, tuberculosis detection and antibiotic resistant testing. नेक्स्ट अगर हम इसके एडवांटेजेस की बात करें, PCR offers several advantages including its speed, sensitivity, specificity. Rapid PCR can amplify the DNA sequence within the hour, allowing for quick test result. PCR can detect very low level of DNA, making it highly sensitive. Specificity की बात करें, PCR can amplify specific target sequences, reducing the chance of error. नेक्स्ट अगर हम इसकी लिमिटेशंस की बात करें, Contamination. PCR is prone to contamination, which can lead to false result. Amplification by some sequences may be preferentially amplified over others. Inhibitors. PCR can inhibited by substances present in the sample affecting amplification efficiency. Future of PCR. Advancement in PCR technology will improve on its sensitivity, speed, and efficiency. Then integration of PCR with other technologies like next generation sequencing will further enhance its capabilities in molecular diagnostic. In coming year, PCR is expected to remain cornerstone in molecular biology research and clinical application. That's all about my topic. Thank you so much for watching.